Cholesterol and fatty acids, yeah. Okay, before we get started, let me show you guys something. Make room. Traffic jam. We usually have very little physiology tutoring, but they have invested in physiology tutoring this time. So I want you to know that in the STEM Center, and I will post this on melanieb.com and on SLCC Phys, in the STEM Center, look at all this phys tutoring that they have, right? Yeah?
Yes.
Not that one. That one's only anatomy. But the rest of this is all phys tutoring. Here's what I might do with that on this campus. Here's what I might do with that. If you have a study buddy, go study there. Because then, if you need help, there's somebody there who can help you, right? I mean, if you're already on campus and you were already going to study anyway, you can go there. And Andy is tutoring, I think Jane and Suzette, and they're all really bright. They're all anatomy lab aides too. Huh?
Amanda?
Yes.
No, not
the other Amanda.
I don't know if Amanda is tutoring for phys or not, but anyway, not only are they tutoring, but they're also pretty good at teaching because they've been lab aides. So it's a decent time investment. Andy has all my notes, and he's already looked through all of them and kind of knows how I do things. So he might be a good one too. Just wanted to tell you that. Second thing I want to tell you is that you are supposed to have your first test next Thursday, but somehow I got a little behind, so it's going to be the Tuesday after.
Yes. So
I'm moving it one time. Okay. If you were busy that day, get unbusy because you're supposed to be here. Okay. So I don't know why I'm behind. I'm supposed to be doing membrane potentials, but I won't get to them until next time. What? Yeah, I'm recording. Okay, and then so this set of notes is the last one that's really got a ton to do with 1610, a ton of overlap. Then after that, we'll get into body systems, and we'll actually start with endocrine system, which is one of my favorites. It's also, fair warning, one of the hardest ones. Okay, so don't miss endocrine lectures. Bad plan. Don't miss them. So what we're going to do in this chapter is stuff that you've done before, but with a different spin. So you've learned about channels and carriers before, and you've learned about osmosis and active transport. This time, don't listen to the what. Your objective is to get to the point that you can predict how you would transport a molecule without me telling you. So what I'm going to do, instead of just saying this is a channel, I will say these are the things that travel through channels. And then you'll know, okay, I'm going to meet a new thing. I'm going to figure out whether that will go through a channel or a carrier or do simple diffusion, or whether it would need to do endo or exocytosis. So what you're looking for here is gaining the ability to predict what's going to be the next time. And then at the end of this set of notes, we're actually going to apply it to human physiology. So instead of just talking about cell transport, we are going to try to figure out how you absorb glucose and amino acids of the small intestine, yeah? How you absorb water, how you absorb sodium, right? But instead of me telling you, based on what I taught you, you're going to say, oh, glucose is of this, we need to absorb it this way across the apical surface, but then across the basal surface, it's got a different concentration gradient, so maybe I have to do it a different way, and then how the hell do I get it into the bloodstream, right? So this is just the last little bit of cell before we really get into like body systems physiology. And why do I have to do this when you do it in 1610? Because people memorize things in 1610 and very rarely do they actually understand them. But in phys you have to understand or else you can't use it for the next time. So let's start with here. Stuff that you've heard a million times before. You know, of course, that the plasma membrane is the boundary between the ICF and the ECF, right? And you know that it's selectively permeable, but it's pretty consistent in its structure. Of course, it's made of a phospholipid bilayer with embedded proteins. That's only showing you channels, but of course you have carriers, you have receptors and all that kind of stuff. But, so what is it that determines how something traverses the cell membrane? If the cell membrane is pretty consistent, then it must be the characteristics of the solute, the thing trying to get through, yeah? That impacts how it travels. You guys with me? If cell membranes are cell membranes, then the difference between how glucose travels and how oxygen travels has to do with the difference between glucose and oxygen. Yeah? Okay, so what we are going to do today is we're going to say, okay, you've been told 1,000,000 times that the plasma membrane is selectively permeable, but how is the selection done, and can you start to predict it? You with me? Okay, so of course it's selectively permeable. The permeability of the substance across the plasma membrane has primarily to do with the chemistry, the charge, the size of the solute, the thing that's trying to get across. You guys with me? Okay, so predict for me. If we have ICF and ECF, first thing is just predict for me which is going to more easily travel in and out of the cell, a large or a small solute.
Small.
Yes, smaller solutes travel more easily across the cell membrane as a general rule, right? So if I'm like absorbing an entire cell, it's going to be way more problematic, a little harder, probably more energy consuming in the process. But if it's something tiny, I might be able to get it across. All right, so that's good. And then the second thing is, you may or may not remember this from 1610, but the charged nature of the molecule, the polarity of the molecule, or the electrical charge, will impact its solubility. Do you guys remember what polarity implies? What does that mean?
Charge.
Yeah, so if you have a polar molecule, it tends to have negative charge on one end and a positive charge on the other. Of course, ions are even more strongly charged than that, right? Sodium has a straight positive charge because it lost an electron, okay? Now, next question is, polar and charged molecules, do those tend to be hydrophilic or hydrophobic?
No,
Hydrophilic, so they like water. Anything that likes water tends to not like lipids. You with me so far? If you are hydrophilic, you tend to be lipophobic. Okay, now, that means that when I run into something like sodium, which is highly charged, yes? Yeah, sodium ion? Can it go straight through the lipid portion of the plasma membrane? No, it cannot. Charged molecule is hydrophilic, lipophobic. Got it? Okay. Now, molecules that are charged do not traverse easily through the plasma membrane, but that doesn't mean that you can't get them in. So we will come back to those in just a second, but generally speaking, something small and charged like that can't go straight through the plasma membrane, but we will see that quite often they can get in and out via channels. Yeah? Channels are relatively small. They don't let big things through, right? Okay, so this might be a sodium channel, and we're going to come back to that. Now, conceptually, I want you guys to start thinking about the cell. Whenever something is going through the plasma membrane and you're transporting it, the cell is going to go cheapest, fastest way first. Cheapest, fastest way first. So think about that, and I'm going to ask you guys a couple questions as we go through. So next thing, so the electrical charge or polarity is going to force it to go through here instead of through anywhere else. Now, then after we finish dealing with electrical charge, we can go more broadly to the concept of solubility. Lipid-soluble substances, nonpolar molecules like oxygen gas, do those have any trouble getting in and out of the cell?
No.
No. So oxygen gas can go straight in and straight out based primarily on its concentration gradient, right? It doesn't have to ask permission to do it, right? You can't even keep it from doing that, as a matter of fact. It will diffuse down its concentration gradient. And that matters, of course. One of the things that it does is it makes oxygen really, really easy to get into the cell whenever you get it near the cell, correct? You get it into the extracellular fluid, it just moves straight into the cell. You don't have to worry about transporting it. But of course, lipid-soluble substances, also it makes it hard for them to stay in the cell if they're lipid-soluble. So for instance, if you make a lipid-soluble hormone like estrogen or cortisol or aldosterone or testosterone, it's lipid-soluble, do you store it? No, you make it release it, make it release it, make it release it, make it release it, because it's lipid-soluble, right? So you can't really keep it sequestered inside the cell. And then also it matters to you sometimes because you depend on that solubility. Alcohol, both topical alcohol and ingested alcohol, is lipid soluble. So if you are on an empty stomach over 21 and do not have cultural limitations, let's engage in this process. But if I take a sip of wine, how long is it on an empty stomach before I actually feel the impacts? And are not pregnant, by the way. Yeah, it's
Like,
just
that's
as soon as it actually circulates to my brain that I actually feel the impacts. And why is that? Because it's lipid soluble, right? There's no impediment to transport, okay? So now I want you to back up and think about these two instances. Which of these two is going to be faster? Simple diffusion straight through the cell membrane or diffusion through a channel?
Simple diffusion.
Now explain to me why. You're right, but explain to me why.
More access points.
Exactly, more access points. One door versus the whole damn thing, right? Okay, so here's what I'm going to ask you multiple times today. If you diffuse something through a channel, I'm going to ask you why you didn't use simple diffusion, right? And then after we get to carriers in just a second, if you use a carrier, I'm going to ask you why you didn't use a channel and why you didn't use simple diffusion. Because cheapest, fastest way first. You with me? Okay, so lipid-soluble substances, nonpolar, fatty acids move straight in. We don't have to worry about transporting those inside the cell. If I had fatty acids in my bloodstream, they get into my extracellular fluid. They go straight into the cell. I don't have to worry about insulin or anything, and you can use them for cellular respiration. Good? Okay, now let's go to water-soluble substances. If you are a water-soluble substance and you are tiny, you might be able to sneak, but there's an asterisk on that. So when I took phys way back when, we didn't know about aquaporins. Do you guys know about aquaporins? Okay, so water is obviously water soluble. Yeah? So we got that one, right? And so you guys know from osmosis that water tends to move in and out of cells down its concentration gradient. And we thought, oh, well, it's water soluble, so it shouldn't do that. So what the hell's going on? Oh, we thought, oh, it's just small enough that it does it, right? There's actually a whole bunch of channels. So water moves in and out based on its concentration gradient. So does carbon dioxide. Carbon dioxide is water soluble. It is polar. It shouldn't. I do not know this to be true, but I wonder whether there are a whole bunch of carbon dioxide channels too, right? Like there are aquaporins water channels. So regardless, these things are relatively small, carbon dioxide, urea, a few others. They somehow break the rules and get straight through the plasma membrane. We know why water does now, but we don't know why the other ones do, okay? If you're really small and water soluble, you might be able to have a caveat and get through. But now let's move up to mid-sized water-soluble molecules. What do I mean by mid-sized? H2O is 3 atoms. CO2 is 3 atoms. This is a mid-sized. C6, H12, 6. Mid-sized water-soluble, right? And you know that sugars are water-soluble, right? Okay. So how do I get those in? So let's think about this through, okay? Can I diffuse it straight through the plasma membrane? Why not? Too big and water soluble, yes? Can I move it through a channel? No, because often it is bigger than the channel, okay? Not always, but often it is bigger than the channel. So plan C is I will move it through a carrier, yeah? So what is a carrier? You with me? Okay, so let's look at what a carrier is. So you can see a glucose carrier here. A carrier is a cell membrane protein. It has a shape-specific binding site for the thing that it's going to transport. And notice that it binds the transported molecule, right? The binding to the carrier changes the shape of the carrier so that you dump the molecule. The dumping of the molecule changes the shape of the carrier so that the binding site is exposed again. Okay. People are in the habit of thinking that that always requires ATP. It does not. If glucose was high on the top and low on the bottom, this occurs in the absence of ATP, right? This is just called facilitated diffusion, diffusion through a carrier. It doesn't require ATP, okay? Is that okay? If you were trying to move glucose against its concentration gradient, then it would require ATP. Okay, so now let's talk about these again. Let's call this one A, this one B, and this one C. So that's supposed to be a carrier. Which one of these is the very fastest? A. And which one's the very slowest? C, right? Even though they're all cheap, right? Free. C is slower. Somebody explain to me in your words why C is the slowest one.
Before it can go through, it actually has to bind.
Has to bind, change shape, right? Dump, change shape again, right? So it's going to be even slower than a channel, right? So the reason I say that is because you would never do C if you could do B or A, right? So if you are using a carrier, you better be able to defend why you weren't using a channel or simple diffusion. Does that make sense? Okay, so glucose, amino acids, those are mid-sized water-soluble molecules. That's what I'm talking about when I say that. Now, once you get it to something really big that still needs to get out, like a big protein. Insulin is made inside the pancreas, but it doesn't do you a damn bit of good if it stays there, right? It's a relatively large protein. It's not huge, but it's pretty big, okay? But I still need to get it out of the cell into the extracellular fluid and into the bloodstream. You with me? Can I diffuse it straight up? I can't. Why? Too big, and it's not lipid soluble. Okay, it's water soluble, not lipid soluble. Can I move it through a channel? No, why not? It's too big, yeah. Okay, it's bigger than the channel. Can I move it through a carrier? Bigger than the carrier. So what is plan B? The slowest, most energetically expensive transport mechanism is endo and exocytosis, right? They're really slow, they're really energetically expensive. You would never do them if there was another option. Does that make sense? Okay, so you can get stuff in and out. It's going to be slow and it's going to be expensive. Okay, so when you are attempting to transport a substance, you always want to go through this mentally to figure out why you're transporting it the way that you are, and not memorize that I told you that glucose uses a carrier, but think about why does glucose use a carrier, right? That makes sense? Okay, so we'll come back and label these in just a minute because I want to teach you a couple other things. All right, so now let's talk about simple diffusion. And you've learned this a million times. I just want you to be able to figure out in what circumstances you would use it. So with simple diffusion, basically what we have is if we have a situation in which I have a high concentration in one location and a low concentration in the other, I will do simple diffusion. So predict for me. This is a concentration gradient. And this is flux, which is just really the transport rate. You with me? Okay. If I do simple diffusion and I start out like this, and I just give it a minute, diffusion, what is this graph going to look like? What's it going to do? Yeah, it's just going to do that, right? Because all diffusion is basically those molecules smack into one another until they all have equal space. If one of those, this little guy right here, was called George, does George have the autonomy to know that he's supposed to go to the other side? No, it's completely random, right? George is just as likely to stay on one side as to end up on the other side because it's not a directed movement of individual molecules. It's just a net, right, movement, net flux. Does that make sense to you guys? So this is simple diffusion. Now, simple diffusion could or could not involve a membrane. If it involved a membrane, then the membrane has to be permeable to the solute, right? So if there's a membrane right here and I am George, right, you guys can come over to my side if you are George soluble, right? If the membrane is George soluble, right? Does that make sense? So if all of us are George, you can come over. I wouldn't go over there because there's a higher concentration over there. Does that make sense? So it can happen through a membrane if the membrane is soluble to the solute, right? Does that make sense? Okay. So what ends up happening is you have a net directed movement in one direction, but each individual particle doesn't know which direction it's going to go. So I want to talk about what is necessary for simple diffusion. It's passive. It doesn't require ATP at all, okay? Just moves down a concentration gradient, and primarily it's just stuff smashing into one another. It depends on the concentration gradient. The net movement is from high to low, but each individual particle is not directed. Distance does matter, so let's talk about that. Diffusion works really well over microscopic distances, but it's really inefficient over long distances. So for instance, when I breathe in, oxygen moves from the alveoli to my capillaries by diffusion. Yes? You with me? microscopically separated. But I need it to go from the capillaries here to my brain, and that's a much longer distance. Is diffusion going to be an effective transport mechanism over those distances? No, right? So it's really a really effective transport mechanism over microscopic distances, and then when you're going much longer, you're going to need something to help out diffusion, right? You can't just wait for it to get there because your brain would be dead, okay? So distance does matter. The diffusion may or may not involve a membrane. Like I said, it has to be permeable. If it is going through a membrane, then the amount of membrane that you have matters. So let's go just a hypothetical and say, okay, here's the bloodstream, and let's make this a fenestrated capillary, and I want to talk about transport in two different cells. So let's say I've got this cell, and I've got this cell, and I'm going to call them A and B. And what I'm trying to do is to absorb something and get it into the bloodstream. Which one's going to have a faster absorption rate? B, for simple diffusion, right? It just has more surface area. That's microvilli, right? That's why you have microvilli in the cells lining your small intestine, because it increases transport rate. Does that make sense? So that matters as well. Is that okay? Okay. So the effect of an increased surface area if you're moving something through a membrane is that it increases transport rate, right? Okay. Now, diffusion works really, really well in fluids, and it also works pretty well in air, right? If there are things that can float in the air, you know that you can have diffusion. So let's talk about a couple of different situations. When I was a kid, we drank Kool-Aid. They don't make Kool-Aid that much anymore because how much sugar do you put in a pitcher of Kool-Aid? 3 cups. No, it's one. Nick, it is 1 cup.
Kidding.
3 cups.
You're making more than one packet than you have.
So, and you of course make fruit punch because that's the only good kind, right? So when I was a kid, and it was off-brand Kool-Aid because we were po. So off-brand Kool-Aid, right? Drink-a-made, something, in a cup, right? And you put the Kool-Aid in there. And let's say you got really distracted, SpongeBob was on or something, and then you don't stir it, and then you come back in the kitchen like 2 hours later. Is most of the water going to be red? It totally is. And why is that? Because it went from high red Kool-Aid concentration to low red Kool-Aid concentration. And is most of the water going to be kind of sweet? It's going to be pretty sweet, right? If you had a couple weeks to wait, you wouldn't even need to stir it, right? They'd be growing mold on the top, but you wouldn't need to stir it at all. So it works relatively well without putting any energy into it, right? Also works in air with things that can travel in the air, and you've experienced this if you have a family and have had a car ride and somebody farted, because somebody always does, in your car ride, right? Especially if you have children. And they do it on purpose, the little bastards. Use birth control, that's all I'm saying. So, they fart in your car, and it starts with your 4-year-old that has a high concentration of stink right near his booty, right? And then you're up there minding your own damn business, driving him all over to his lessons, right? And then what happens? It moves from high stink concentration to low stink concentration and shares the love all over the car, right? And that is diffusion. And it would work even if there was a membrane, if the membrane was permeable to the stink. So if you had one of those dog gates, don't put your kids behind the dog gate, okay? But if you had one, right, just don't. There's no car seats back there. If you had one of those dog gates and your dog farted, you would still smell it, right? Because the gate was permeable to stink. Yes? Okay, got it? Because it was permeable to stink. Okay, so now, just to tell you guys, I'm not going to get into this today, but I do want to acknowledge that there is another thing besides concentration gradient that can motivate diffusion. And I'm not going to go into it in a lot of detail today, but I do want to tell you just this. I want you to think about a couple of different situations. I've got sodium, high concentration, and there's a channel to move it through because it needs one, okay? Okay. If you are moving a charged particle, quite often it needs a channel. It's still diffusion, but it's diffusion through a channel. But it's not just concentration that will motivate ions to move. Because if I say it's high here and low here, yes, then we all know it's going to move in. But I can impact the speed with the charge. So you tell me which one, again, A versus B, which one is going to move faster? If the inside of the cell has a slight negative charge, or if the inside of the cell has a slight positive charge. Yeah, so ions move due to concentration and charge. Yeah? Okay, and collectively we will talk about that an awful lot this semester. We call that collectively not their concentration gradient, but their electrochemical gradient. Yes? Electrochemical gradient. Okay, so other things that are not charged don't have to worry about how charge would impact their movement. All right, so now, when diffusion can't occur directly through the cell membrane, it can and does occur through a channel protein. And here's one of the types of channel proteins. All a channel protein is a transmembrane protein, right, that allows for a space for something that wouldn't otherwise to get through. And like if I draw this in here and call this a sodium channel, I'm telling you that it likes sodium really well, but it's not specific to sodium because it doesn't have a binding site. So it's sodium is my favorite, but maybe another positive ion would kind of do channel, but we don't call it that. We just call it a sodium channel, right? And right, exactly. But without a shape-specific binding site, does it make sense to you that it might move almost any positive ion that was sodium size or smaller, right? But if it moves sodium most of the time, we call it a sodium channel. I just need you to understand that that's going to be different than a carrier because a carrier is super specific about what it moves. Good? Okay, so you can move through channels. Ions do quite often move through channels, okay? There are 4 different categories of channels. The one that you see right here is always open and just allows ions to move in and out down their gradients, right? We call this a leak channel, okay? So now let's label this one a sodium leak channel. Leak channels are always open, okay? Can't close them. So we will see these quite often. It's a little difficult at first to figure out why the hell they exist because they just seem to be jacking things up. We will figure out later that they're really important. It just looks like they're messing everything up. So that's a leak channel, always open. But then the next category of channels are called gated channels, okay? And gated channels have 2 different states, open and closed, okay? So let me show you what a gated channel would look like in its closed state. This is a gated channel closed. This is a gated channel open, right? Once it's open, right? But before it's open, nothing gets through it. So let's talk about how you can open and close gated channels. You ready? So there's 3 different kinds of gated channels. This one is called a ligand gated channel, and it will open when a ligand binds to it. Yeah? So how do you open that channel? You bind a ligand to it, and that's what makes it open. Guess what? Those ligands that open ligand-gated channels are usually intercellular chemical messengers. Like, give me some examples. What are some examples of intercellular chemical messengers? What'd you say, Andrew? Neurotransmitters, fantastic. Give me some more. Come on. Intercellular chemical messengers. We spent a long time on. Paraffin agents. Autocrine agents. Hormones. Yes. So one of the many things that you can tell another cell to do, if I am the releasing cell and you are my target cell, I can release something that says open a sodium channel, and then sodium will move into the cell, right? That's one of the many things that will happen. Is that okay? Okay. So often intercellular chemical messengers. So this is a ligand-gated ion channel, right? A ligand-gated ion channel. And then the next one, I have no idea how to draw it. I've got it in your notes, but I don't know how to draw it. So I'll just tell you how it works, and we will get into them, and I'll show you videos of them later. But the next type of gated channel is called a voltage-gated channel. And so what I want you to imagine is that this one does not open when you bind a ligand to it, but when you zap it, right? When you zap it with electricity. So think about this. with a person getting electrocuted. Which body systems are dramatically impacted when you're electrocuted? Cardiac muscle, nervous system, skeletal muscle, smooth muscle, because you usually poop yourself, right? Okay, so all of those electrical systems in the body have voltage-gated channels. Voltage-gated channels. It is going to take me a minute to show you guys how you actually generate voltage within the body. So that's a lecture for another time. But just let me tell you that there are ion channels that are opened or closed by changes in voltage. Okay? And we will get it, and you'll actually understand it, but it takes a while to build to it. So there are voltage-gated channels. You're going to only find those in your electrical systems. So which body systems have you always learned are electrical? Nervous system, cardiac muscle, skeletal muscle, smooth muscle. All of those. Go ahead. What was your question?
Just going back to the ligand.
Uh-huh.
Will you always call them ligand-gated, or will you kind of switch it between?
Well, I learned chemicals, so sometimes I say that, but I'm trying to be consistent with what your textbook does. So I'm trying to say ligand-gated channels, but since I learned it as chemical-gated channels, sometimes that'll come out, right? So they do open by chemicals, but I'm trying to switch to ligand. Okay, and then last but not least we don't talk about these very much this semester. They're interesting though. Mechanical gated channels, okay. These are channels that open and close when you stretch them or squish them. So I want you to think about this example for a mechanical gated channel. So if you pull on it or squish on it, it'll open. So I want you to imagine that you fell asleep and you woke up and it was pitch dark, and your arm was right here, and you couldn't see it, right? And you didn't remember putting it there because you were asleep. So here's my question. How do you know your arm is there? You know because channels in this ligament and tendon here are stretched, and they send some signal about the position, and these are squished, and they send another signal about the body position, yeah? Because you are opening and closing certain channels that help you with what we call proprioception, which is how you figure out where your body is in space. Does that make sense? Okay. Really, really important for positioning your body in space. There's a lot of that goes on with these mechanical gated channels. Everybody okay with that? Okay. So, yeah, we did carriers. Okay, so let's go back here and see if you guys can do just from this figure that's in your textbook or in your notes. Which one of these is a leak channel? Which letter? A is a leak channel, yeah? What is B? It's two different states of a what channel? Please note the binding site up here. Yeah, those are two different states of a ligand-gated channel, closed and open. Yeah, good. What do you think that D is? D and E. Two different states of a carrier, good, okay. And then H and I, those are mechanical gated channels because if you squished them, you could squish those open, you could pull them open. And then these are weird looking ones. That's actually what voltage gated ion channels look like. So that's two different states of a voltage gated channel, F and G are. Cool? And we'll see voltage gated channels in a lot more detail later. Everybody okay? Okay. Mechanical gated channels, open and closed. Okay, so before we go any further, and we're going to do osmosis, you've learned it a million times, but this time you actually have to understand it. Before we go any further, I want to see if you guys can distinguish between this one and this one for me. So what is the difference between a carrier and a ligand-gated ion channel? What's the difference?
The ligand gated doesn't have a binding site, and then the
It doesn't have a what?
It has a binding site.
They both have binding
Oh, they
sites.
both have binding sites.
But what's the difference? See if you can figure it out.
One like just has the door that opens,
Mhm.
allows multiple ones to go through, and the other one can only one at a time, then change.
Right, and then also let's think about what's transported. Do you transport the thing that you bind here? No, and you only transport the thing that you bind there, right? So even though both of them have shape-specific binding sites, they are not transporting the same thing. This is just the mechanism of opening this one. Once it's open, it kind of acts like a leak channel until you close it again, right? Okay, but this one is transporting the thing that binds to it. So it's very specific about what it transports, right? Okay, and do you think this one is faster or slower than that one? It's going to be way slower, right? It's going to be way slower transport mechanism. So you cannot use the terms channel and carrier interchangeably. And I know people get into the habit of doing it, but I'm going to have you correct yourself every time you say it incorrectly because they don't mean the same thing and they don't have the same physiology. Go ahead.
So with carriers,
Yes.
you said they can allow for multiple things to be into the intracellular?
No,
Or
they
just
bind one
once?
thing.
Okay.
Oh, they don't work once, but they work once at a time, right? You bind it, you dump it, you bind it, you dump it, right? So it's like things are just waiting in line to go through that one. Does that make sense? Okay. Everybody all right with that? Okay. All right. So now let us talk about osmosis, but we have to talk about osmosis in a you truly understand it way because I know that ever since I was in 7th grade, I've learned osmosis, and mostly what people do is memorize it, but they don't get it. And then when I try at the kidney to teach you the countercurrent multiplier mechanism to show you how the kidney concentrates urine and actually pulls more water into the body, if you memorized osmosis and didn't understand it, you have no idea what the hell's going on, okay? So we have to actually, I want you guys to be honest with yourselves and honest with me. And if you don't get what I'm saying, it is important that you stop me this time, okay? All right, so what we are going to do is we're going to first define osmosis. And the first thing that you need to know about osmosis is really, it's just a special instance of diffusion. And by special instance, I mean it's something very specific. So it is only diffusion of water, ever. And it's always across a selectively permeable membrane. Diffusion of water across a selectively permeable membrane. So it's passive because all diffusion is passive. Doesn't require ATP, right? And it depends upon a concentration gradient, but you're not usually given the relevant concentration gradient. So let me show you, and then we're going to kind of draw it. Okay, so this is an osmosis situation. And notice that they give you the solute concentration and the water concentration. But are you ever, any other time, given the water concentration? No. They just give you the solute concentration, but then you don't move the solute. And so you have to figure out what the heck you're doing here. So let's build it and figure it out. So if we build an osmosis situation, we're going to be good scientists, and we're only going to vary a single variable at a time and hold everything else constant. So we're going to get 3 situations right here, and we're going to put identical cells in each beaker, all with selectively permeable membranes. Okay, they're all going to have the same quantity of solutes in them. Cells are pretty similar. So X is going to be a solute. Okay. And then I'm going to vary the extracellular fluid. So you guys tell me, which one do you think is more likely to vary anyway? Intracellular fluid or extracellular fluid? Which one's more likely to vary? Extracellular fluid is way more likely to vary, so we're going to vary it. So we're going to call these situations A, B, and C. And then what I'm going to do is I'm going to vary the concentration of solutes in each one. In A, no solutes, right? Pure water. In B, I'm going to try to balance the concentration inside with the concentration outside. And then in C, I'm going to put it in a really, really concentrated solution. Now, the important thing for you to know that will tell you to think about osmosis is that this solute has to be non-penetrating or impermeable. What does that mean? It can't cross the cell membrane. If you hear non-penetrating or impermeable solute, that pricks up your ears and says osmosis, not diffusion. Does that make sense? Because it can't move. If the solute could move, then we wouldn't be doing osmosis, right? Okay, so let me show you a video, and then we will actually figure out why all of this happens. No, not that one. Where did my video go? I lost it. Oh, there it is. Okay, ready? So, blood cells. We're going to do this exact experiment, and you already kind of know this. So you're actually just seeing a whole bunch of red blood cells in a capillary. Then we're going to do a different experiment. We're going to put blood cells in an isotonic solution, which is your normal saline. What should happen to the size and shape of those blood cells? Nothing. They should stay pretty much the same. Then we're going to put it in a hypotonic solution. What should happen to the size and shape of those cells? They will become spheres, right? And if you let them go for long enough, they will actually do lysis, right? Okay. And then we're going to put those same cells in a highly concentrated solution. And then what will happen to these cells? They're going to shrink. Okay, so you guys have known that for a while, but what I want you guys to be able to do is to explain to me why as we're going through this. So let's go back to this, talk about it, and say, okay. Okay, so what I have is non-penetrating solutes or impermeable solutes. I've got water, and I've got a selectively permeable membrane. So in this situation, water is the only thing that can move. Okay, and water diffuses down water's concentration gradient. Okay, the way a lot of you guys have it memorized is that water moves toward high solute concentrations, which is what happens, but not why. And it will not help you once you get to the kidney, because water really moves down water's concentration gradient, right? So water moves from high water concentration to low water concentration. It is not moving toward high solute concentration because it doesn't give a crap about the solutes. It's moving down its own concentration gradient. So I'm going to ask you the same questions in all three of these situations. So in this situation, first question is, which direction will water move? Which direction? In or out of the cell?
It's going to stay the same.
No.
It's out.
In. Why? Somebody explain. More water on the outside because there's solutes on the inside, right? That makes sense? So there's a lower water concentration comparatively on the inside, right? Pure water on the outside, not pure water on the inside, right? So what will this do to the size and shape of this cell? It will swell. And if it swells big enough, it will cause lysis. If it's a red blood cell, you call it hemolysis. Good? Okay. That doesn't happen to a plant, though. I can pour pure water on a plant, and the cells will swell, and it will stand up nice and proud. Why doesn't it go through lysis? What do plants have that you don't have? Cell wall. Exactly. Yes. And you are putting a pure water solution on them, and they fill up, makes them stand up. Good? But you don't have a cell wall. Okay? All right. So in this situation, again, same questions. Which direction is water going to move?
Either way.
It's not gonna run anywhere.
It's bound.
Does it really go nowhere, or does it go net nowhere?
Net.
Net nowhere, right? But you can't make it impermeable to water, but it goes in and out at about the same rate, right? So what does that do to the cell? Same shape, same size, same. Even if I didn't draw it the same, that's what I'm trying to. Is that okay? Okay. And then this situation, same two questions. Which direction is water going to move? Out. Somebody who has not spoken yet, please verbalize to me why water moves out. Because there's comparatively less water on the outside, right? Does that make sense? Because it's a highly concentrated solute solution, right? Does that make sense?
Yeah.
Got it? So water moves down water's concentration gradient. Okay? So if there is less water, it's going to move to less water. Good? It doesn't move to high solute concentrations. So if you have two different solutions, then water doesn't care what's in either solution except for the concentration of water. Yes? And you guys are going to do osmosis and diffusion in the lab, and you have to, if water's moving, water only cares about water. Okay? Doesn't care about solutes. What did you have a question, Andrea? Was that a hand raise or no? Okay. All right. So everybody okay with that? Okay. So go ahead.
Does this have like anything to do with homeostasis in your body? Or does
Totally.
water not care?
No, absolutely has to do with homeostasis in the body. The kidney is constantly comparing the water concentration of your blood plasma to the water concentration of your urine and comparing water concentration to figure out which way it should go. Sometimes I'm overhydrated and I need to dump some into my urine, right? And sometimes I'm always dehydrated because I pretty much live on coffee until it's time to have a cocktail. So I'm always dehydrated. So it's always comparing those two and saying save everything that you can, right? So it's comparing these two solutions, and then water moves down water's concentration gradient at the kidneys. Good? In order to maintain homeostasis. So yes, it absolutely has to do with homeostasis. And that's why I can't have you memorize it. I need to have you understand it because you're going to have to compare two different solutions. Okay, so we're going to talk about some examples in the human body. But first, let's name these solutions. You've done it before. Oh, sorry, with C. What happens to the size and shape of this cell?
It shrinks.
It shrinks. What's that called? Crenation. Okay, now all we have to do is label these solutions. Note, you are labeling the solution with reference to the cell. The cell's the standard. It doesn't change very much. And you are going to use prefixes that are familiar to you. One of these solutions, A, B, or C, has about the same amount of solutes as the cell, and so it gets the prefix that means same. Which one is it? B. And this is an isotonic solution. Good? Okay. One of these has greater solutes in the extracellular fluid than in the cell, and it gets the prefix that means above or more than. Hyper, which one?
C.
C. One of these has fewer solutes than the cell, so it gets the prefix that means below, which is hypo and hypotonic. Okay? How I remember this from 7th grade when I learned it is if you're hyper, do you get skinny or fat? You get skinny. And then hypo, hippo, big fat cell. And then that one.
ISO, you're the same.
What?
You're ISO.
ISO. You don't have to remember ISO if you got the other two, right? Like hypo, hippo, big fat cell. Okay, so let's talk about a couple of examples in human physiology of these situations. So, get dehydrated in the middle of the summer, right? People do it all the time. You go into the hospital, and they want to replenish you. Do they shoot you up with straight water? No, what do they shoot you up with? Normal saline, which is usually 0.9% because it is blank tonic. Isotonic to your blood cells. Yes? Okay, because your cells aren't empty, so you can't have the solution be empty, otherwise water would move. Does that make sense? Yes? Okay. Physiology is all about aha moments, man. That's what it's about. Okay, now, I want to talk about now why, if you're stranded on a desert island, you should not drink salt water. Why should you not drink salt water? I'm going to drink some salt water because I didn't take physiology. And I want to know what's going to happen to all of those epithelial cells in my digestive tract as the salt water goes by. What is going to happen? You're drinking a hypertonic solution. What is it going to do? It's going to suck the fluid out of every cell that it comes in contact with, yes? And then what you are going to do is you are going to have the squirts, diarrhea, right? Does that make sense? And you are going to poop out and pee out everything that it touched, and you're going to end up with less fluid than you started with. You are going to end up losing your intracellular fluid into your urine and your poop. Does that make sense? So if you get stranded on a desert island, you definitely do not drink salt water. You try really hard to find some fresh water by any means necessary. And then if absolutely necessary, you can drink your own pee. It's better than actually drinking salt water, but make sure it's yours. Because you got some weird bugs in there, but you're already used to them, right? Pee is mostly sterile, but not exclusively. Once it goes out the urethra, it ceases to be sterile. When it's in the urinary bladder, it's usually sterile unless you have an infection, right?
So why are you not supposed to eat snow? Is it, like a lot of people say don't eat snow because
It doesn't dehydrate.
Really don't eat yellow snow.
like,
It's just crunchy water.
Okay, that's what I thought, but
I think
like,
you're referring to people saying that in like extreme survival situations where you're gonna lose body temperature.
Oh, yeah. So you're
That's
if
why
you're
they
just,
say don't eat snow.
yeah.
You're already
Okay.
cold.
So it's more about body temperature regulation
Than
than
it is.
water regulation.
Okay.
Yeah.
Makes sense.
And then last but not least, hypotonic solution. This one's counterintuitive. So physiology situation in which you have a actually I've got two. One of them is what would happen if I were dehydrated and I went into the hospital and they shot me up with straight water.
And all your cells are gonna
My cells would swell and potentially lyse. But first my kidneys would try really hard to compensate, right?
Mhm.
Okay, my kidneys would try super hard to pee out all the excess water. If I overwhelmed the capacity of my kidneys to do that, then I could have these problems. Okay, now there is a tragic situation that happened like, I don't know, 6 years ago, maybe more, here in Utah. Did you guys hear about this? About this family that decided to punish their 6-year-old daughter-ish,
And
I
by
think.
drinking water,
By drinking.
a gallon of
By
water.
making, so she drank something out of the fridge that she wasn't supposed to drink, like one of Dad's sodas or something. And so they decided to punish her by having her drink gallons of water. Gallons of water. And she overwhelmed her kidneys' capacity to regulate her water balance. She ended up with hypotonic blood plasma. But the problem wasn't the impact on her red blood cells. The problem was the impact on her brain, because she was actually pumping hypotonic solution near her brain, and her brain cells swelled. And she died. Yeah, she died related to that. So the next time, if you're a parent and you ever think about force feeding or force drinking your child anything, save up for therapy, because they're going to be pissed about it, right? If it's unnecessary, that is not a punishment. That's terrible. I still hate my dad over that sweet and sour pork incident. He made me eat sweet and sour. Oh, okay, my dad was kind of a bully. He's a terrible dad, but he's dead. So it's true. He really wanted to impress the pastor. He's a fundamentalist Christian, really, really like rigid. And the pastor was coming over for dinner, and he's like, for some reason they made sweet and sour pork, which is just neon and gross. And I just hated it. I thought it was gross, and it had green bell peppers in it. I don't eat green bell peppers still, they're gross. But I was with the pastor at the table, so he made me eat it. Just like made me eat it. Made me stay at the table for like 3 hours until I ate it. And right when the pastor was leaving and I was still sitting myself by the table, pastor came up, he made me get up and say goodbye to the pastor, and I bleh, all over his shoes.
How old
I was
were you?
like
That's karma.
7.
You were 7.
Yeah, I was 7. That's all right. He's a bastard. Okay. So I'm a better parent. That's what matters. He did have kids when he was 17, so he didn't know what the hell he was doing. Okay. So now, hypotonic solution, the only other thing that's a hypotonic solution is sometimes interesting is this counterintuitive thing that happens when you soak in a bathtub for a long time. You think your fingers are shrinking, but that doesn't make sense, because what kind of solution are you soaking in? Hypotonic, not hypertonic. Yes, hypotonic. So what your fingers are doing is they are swelling.
You know, like, look.
But do you remember hemidesmosomes that keep the epithelial tissue spot welded every once in a while? So they're swelling except for where they're stuck. And then they're swelling except for where they're stuck. And they're swelling except for where they're stuck. Yeah? So they're actually, yeah, I know. That's totally going to get you a date sometime. You'd be like, I want to tell you about your fingers in the bathtub. And you would be like, we are not alike at all. So that's, and if you had a super duper sensitive scale, you could actually measure how much you weighed, and you would actually weigh slightly more because you would actually absorb some water. Anybody, how many of you guys have kids? Anybody? Yeah, a few. Did your kid, my son freaked out every single time he got out of the tub because he thought his fingers were permanently like that. And he would be like, ah! And I'm like, just chill, just chill, just chill. And then it would go away. And then the next time, ah! I'm like, stop, just stop doing that. It's fine. Yeah, he was like 3. He just like, oh, freaked him out.
You know,
Okay.
I have to look forward to.
So, these are the solutions, right? And again, when you run into these, instead of rote memorization questions, I might set up a situation in which I give you a concentration, yeah? And it might be 300 milliosmoles or 500 milliosmoles. Does it matter that it's milliosmoles?
No.
No, it could be kumquats or elephants because you can compare 300 to 500, right? And you can know, and I won't give you the water concentration because you know you're never given the water concentration. I'll give you the solute concentration, and then I will ask you logic questions instead of memorization questions. Which direction is water going to move? Why? What's the impact on the size of the cell? Name the solution. Good? So those are the kinds of questions that you need. Go ahead.
Are we allowed to have like a scratch paper on our test?
Totally. It can't be your notes. People try that every semester, like, that's all the paper I have. I'm like, I guess you don't have any paper then. Go ahead.
Just to make sure, so water moves in the highest solute concentration.
What?
It moves
You can
towards
say
the
that, but I
less
just
water.
hate it.
Yeah.
Water
Concentration.
moves down water's concentration gradient.
Okay.
Yeah? So water moves from high water to low water. Because is water really aware of solute concentration? No, it's not. It's only moving down its own concentration gradient. Right. But of course I know that that's how people teach it in the past, is
Right.
that water moves toward high solute concentrations. And while it's true, it's not the reason.
So the
Yes?
reason is it moves down its
It moves down its own concentration gradient. Go ahead.
So that's why when you're saying that there's a high solute concentration, the water is moving down that way because there's not a
Low water concentration. Yes. Do you see?
Okay, that makes sense.
Okay. All right. So why does that matter? Well, because a lot of times you'll have two different solutions, both with whatever hell solutes is in there, and maybe different kinds of solutes, but then you will have water, right? And you just look at what water's concentration is, and water will move down its concentration. Does that make sense to you?
Mm-hmm.
So you can ignore which solute it is as long as it's non-penetrating solute, right? Okay, so we will, of course, get into this in detail when we get to the kidney, right? You know you're going to end up talking about it in the kidney. All right, so now we are going to now talk about mediated transport systems. And mediated transport systems use carrier proteins. So we're going to be talking about these. And mediated transport systems are going to be, we'll start with this figure right here. So if you were moving something through a carrier, then the carrier is the mediator. You with me? So when do you need a mediator? Well, when you can't accomplish it yourself. In Utah, if you're going to get a divorce, you have to go through mediation, right? Don't you in Utah? Yeah? Yeah.
I think it's required everywhere.
It's not required everywhere,
Oh, really?
but you have to go through mediation. And what is mediation about? Mediation is about something needs to be accomplished here, but hate your guts. Don't want you to have anything ever. So I've got to have something in the middle that can help us accomplish. The something in the middle is a carrier protein. Yes? Okay. Carrier protein is the mediator. So mediated transport system. So there is some interesting things about mediated transport systems. First off, they always use a carrier protein, and carrier proteins have a shape-specific binding site. And are they slower or faster than channels? Slower than channels, right? But don't necessarily require ATP. We will see some forms of mediated transport do. Okay, so because it has a shape-specific binding site, just stick with me for a second, it is really easy to turn these on and off. Do you remember how you could, via allosteric regulation, turn enzymes on and off? By binding maybe over here, look up here, and changing temporarily the shape of the binding site, right? You can do the same thing with carrier proteins. So this carrier protein moves glucose. I might need insulin to bind to make glucose fit, and then it's on. Yes? Does that make sense? So they're very commonly regulated by other molecules, and the other molecules are usually intercellular chemical messengers. Again, okay? Hormones, neurotransmitters, paracrine, autocrine, right? Okay? So if I just made a glucose binding site on this one, then it would be basically the insulin-mediated glucose transport, right? Okay? Good? Everybody good with that? Okay, so they're really easy because they're shape-specific. Anything shape-specific is really easy to temporarily turn on and off.
So insulin will bind to the.
Not to this site,
Okay.
but to a regulatory site, right? So I want you to imagine for me, because this picture doesn't show it,
Okay.
that it's not glucose-shaped until insulin binds to it, and then it's glucose-shaped.
Get it.
Got it?
Yeah.
Okay?
Insulin has to bind to it.
Yes, not with every, I mean, there's a million different ways to transport glucose because it's super important. But if I'm trying to hook this one together with something that we've already talked about, there is an insulin-mediated glucose carrier. Yes? Okay? And insulin has to bind for glucose to fit. And when insulin falls off, you can't transport any more glucose.
So just a quick
Right?
question. So with the regulatory site, different things can bind to that, and it changes the shape regardless.
Changes the shape of the binding site for the thing you're going to
So
transport.
it can be like this one. Let's say that like right up there, that right here,
Uh-huh.
that's where like the insulin will attach, and then we have another hormone like estrogen or something.
Generally speaking, insulin and estrogen will not bind to the same site. Like, it's shape specific, so it would only fit insulin.
Oh,
There might be another transport mechanism in another cell that binds to estrogen,
So this-
but not on the same
So
cell.
this regulatory site would be only for insulin.
Only for insulin.
Okay, that's what my
Yeah.
question was.
And most carriers are only controlled, if they're controlled by anything, it's controlled by one thing. All right, it just has like one little site that binds to insulin. You okay with that? So carriers, because they're shape specific, are really easy to turn on and off by allosteric regulation, like we talked about before. Okay. And then one other thing about carriers before we get into some of the specifics of it, because it is shape specific, like an enzyme, yeah, it's subject to saturation limits. Okay, so do you remember seeing a picture that was like this for enzymes, but it was concentration gradient and reaction rate? Okay, this one is concentration gradient and net flux. Flux is just the transport rate. So I want to compare a couple things so that you understand this. If this was simple diffusion and not like facilitated diffusion, so facilitated diffusion is from high concentration to low concentration through a carrier, okay? If this was simple diffusion, then I would have concentration gradient and flux, and as the concentration gradient went up, the flux would go up, right? That makes sense? But if you're going through a shape-specific binding site, you set it up the same way. And flux. And as concentration gradient goes up, the flux goes up until what?
Saturation.
But what is saturation? What is happening? What is happening?
So when you have enough on both sides.
No? What happened to cause saturation with enzymes? They're full. They're full, right? If all your carriers are full, it doesn't matter if you dump more glucose in there. They're all waiting in line to get through the glucose carriers that exist, right? So it can get saturated. Yes? What is the only way that you could increase glucose transport after this occurred?
More
Make more carriers, which isn't instantaneous and also doesn't generally happen, FYI. Okay? So these are subject to saturation because they can get full. Are leak channels subject to saturation? No. Right? They're always open. It goes through willy-nilly. Can't actually get full, right? Okay? So you can tell that that's actually through something shape-specific because once you get full, it limits the transport rate. Good? And this will matter. It's going to matter with diabetes and things like that. So, like, you can only reabsorb so much sugar, right? So you end up peeing out a lot of it because at your kidneys, what reabsorbs the glucose back into your bloodstream. So if I was an untreated type 1 diabetic, stick with me, I would have a whole bunch of sugar in my bloodstream, yeah? If I have a whole bunch of sugar in my bloodstream, it gets pushed into my urine, right? I would love to bring it back in because it's money, right? But it's brought back in by carriers. Yes? So there is a limit to how much you can bring back in, so you end up peeing it out even though it's gold. Does that make sense? Okay? Because you're subject to saturation. Yeah? So they just get full and you can't bring it back in. Good? Okay, and we'll come back to that when we do diabetes and the endocrine system, which is next after this. Actually, after this. All right, so carrier protein subject to saturation limits. All right, so now let's just talk about the two different types of transport that we have to deal with. So we have been talking about facilitated diffusion. Facilitated diffusion is just diffusion through a carrier, right? And so if I just told you, again, that glucose was high on top and low on bottom, then this would be an example of facilitated diffusion. It would be moving down its concentration gradient through a carrier, right? Okay, same questions we've been asking before. Why am I moving it through a carrier instead of through a channel? It's too big. Why am I moving it instead of straight through the cell membrane? Too big and water soluble, right? So I'm going to move facilitated diffusion only for things that are too big, wrong solubility to get through the other ways. Okay? This is a common mechanism of moving glucose and amino acids. Okay, and then doesn't require ATP, moves things from high concentration to low concentration. This is how you usually get glucose and amino acids inside your cells, facilitated diffusion. And now we are going to move on to primary active transport. Okay, so now I'm going to be using a carrier still, so it's still mediated, but I'm going to be trying to move something against its concentration gradient, from low to high. So basically uphill. If I'm moving something downhill, do I need to put energy into it to get it to go? No. If I'm moving something uphill, I do, right? So you're going to need ATP if you want to move something against its concentration gradient. So we are going to build to understanding this. Okay, so we're going to be talking about primary active transport first, and next time we'll talk about secondary active transport. With primary active transport, I'm moving ions or molecules against their concentration gradient, and I'm going to need two things. I'm going to need a carrier, and I'm going to need ATP. Carrier plus ATP is called a pump, yeah? And you've seen pumps before, okay? The net movement is from low concentration to high concentration. So with this in mind, you guys said that if I started out and I just did diffusion, right, then it would end up looking like that over time, right? So what I want to understand is if I start out like this and I do active transport, what can it look like? Well, it depends on how quickly you pump it. You can either maintain the concentration gradient you already had, or if you pump it faster, you can accumulate a greater concentration gradient. Does that make sense? Okay. So the metaphor that I like to use for this one is, have you ever seen anybody, or done it yourself, try to fit 24 balloon bouquet into a hatchback? Okay. So you open up the hatchback and you start pushing it in because you're trying to accumulate molecules on one side of the membrane. Yes? But you can't close the hatchback. So as soon as you stop pushing, what do they start doing? They start diffusing straight out. And then some yahoo left the back two windows open, right? So they're going out that way. So if you want to maintain a concentration gradient against what things would like to do, you better work hard at it, right? So you're going to have to put in ATP to maintain the concentration gradient, if the concentration gradient is important to you. And a lot of times it is, okay? So this is called active transport. There's an accumulation of particles on one side of the membrane. The carrier is shape-specific, because all carriers are, and it moves things against its concentration gradient, and it is going to be subject to regulation by other molecules, but we'll get to that in just a second. So I want to spend our last 13 minutes describing to you guys the sodium-potassium pump, but instead of teaching you the pump first, I'm going to show you why the pump exists, okay? So we're going to just start out with some things that are homeostatic for a cell. So first, of course, cells have ICF and ECF. In order to tell this story, all you really need to do is to know concentrations, high-low, not even numbers yet, for sodium and potassium. Does anybody know where sodium is in higher concentration, in the ICF or the ECF? ECF, yes. The ECF has a high concentration of sodium, and that's homeostatic, compared to the ICF, which is a lower concentration of sodium. We'll worry about numbers later, okay? So that's homeostatic, but it's hard to maintain, because as soon as you let it, sodium's going to, of course, leak right in, okay? And potassium, where is potassium high?
Potassium high inside.
Potassium has a high concentration inside the cell and a relatively lower concentration outside the cell. And that's homeostatic, but it's hard to maintain. Everybody good? So, does that mean I never mess with these? No, like everything else that's homeostatic, you need to fix it when you mess with them, right? If it gets jacked up, if your blood pressure goes up to 200/120, you don't die, you bring it back down, because you might have been working out, right? Okay, so this is ideal, but it constantly gets messed with. One of the ways that it constantly gets messed with is that some yahoo left the back two windows open.
Sodium
Okay.
leak channels.
Okay, so let's label these. This one is going to be a sodium leak channel. When given the opportunity, which direction will sodium leak? Out. Why?
Because it's a higher concentration.
Yes, it's leaking down its concentration gradient. Why can't it move through here?
It's not.
Charged. Charged particles do not, they are polar and they don't move through lipids. Okay? Now, this is a potassium leak channel. When given the opportunity, which direction will potassium leak?
Into the cell.
Into the cell. And nope, out of the cell.
Isn't
You guys
that
tell me?
reversed? Isn't
Yeah,
sodium
you guys,
like-
silly. Okay. Sodium leaks in, high to low. Potassium leaks out, high to low. Were you guys testing me? I was testing you and you failed. Okay. Are we okay with that? Okay, so do you see that I have what's homeostatic and then I have a constant challenge? And I know some of you guys are thinking, why the hell are those leak channels there just jacking everything up? Later on, we will figure out that they're really important. But for right now, just trust me that they have to be there. So they create a problem. We actually have a solution to the problem. And the solution, I don't even have to tell you how it works. You guys can tell me. So the solution is a pump, carrier protein plus ATP, that fixes this problem. So I don't have to tell you which direction you should pump sodium. Which direction should you pump it? Homeostasis is your goal. Out. Yes. Why?
Because you're coming in.
Because I want high sodium and I'm jacking with it all the time. Good? Which direction should I pump potassium? In.
Because you want a low concentration on the side.
Exactly. Good? Okay. And to get things to move against their concentration gradients, because they are, right, I need ATP.
And that requires primary active train.
Yeah, ATP to ADP. That's used ATP. Okay? All right? So, this guy right here is the sodium-potassium pump, also sometimes called the sodium-potassium counter-exchange pump because it's going two different ways, or the sodium-potassium ATPase pump. It's all the same pump. You have them on every single cell in your body, and they are so damn important that they gobble up around 40 to 60% of all the ATP that your cell uses. Really, really important. So let's look at this, and then I'm going to ask you guys a couple of questions. So sodium and potassium will, when given the opportunity, diffuse down their concentration gradients through channels, yes? Okay, whenever they're given the opportunity. And we'll put in the electrical nature later, and that's not for today. And it's passive transport, right? But we still have these goals that we need to maintain. So our goals are actually to maintain over time a ratio of about 15 sodiums outside for every one inside, and about 30 potassiums inside for every one outside, okay? And I can maintain this despite these because of this. But how often do I have to use it?
All the time.
All the time. Like as soon as I stop using it, what's going to happen to those sodium potassium concentration gradients? They're going to be level, right? And that won't kill you unless you leave it there. But if you leave it there, that actually is the cell losing its homeostatic control of its ions. Okay? So you can't leave it. No, it's not cancer. Go ahead.
Is it, I could like just be remembering this wrong,
Go ahead.
but like an action potential, and is it like the swapping the like your
Yes, so we're going to do action potentials. One of the things that happen with action potentials, I don't know how many of you guys have actually talked about them. Action potentials completely jack this up big time on purpose to send a really fast signal, and then you have to pump your little buns off to fix it. Okay? It's worth it though, because it's of course the way that your nervous system works, right? But you break it and then you fix it really quickly, as long as you are healthy otherwise. So basically, what I'm doing is I'm maintaining this concentration gradient due to this carrier protein plus ATP, yeah? This pump, carrier protein plus ATP, that allows you to pump things two different directions constantly in order to maintain this. So let's label the types of transport that we see. Let's go A, B, and C. Which of these, A, B, or C, is active transport? C. Why?
Against its concentration.
Against its concentration gradient and is using ATP. And what are A and B?
Passive.
Passive, be more specific.
The passive because it requires no
No,
ATP.
be more specific.
Passive transport.
Be more specific. Passive is general. That includes simple diffusion, diffusion through a carrier, facilitated diffusion. This is diffusion through a channel, right? That makes sense?
Yeah.
Okay. All right. So let's look at the figure from your textbook, and then you can figure this out. Okay, so if we look at this figure right here, and then I have an animation to show you guys as well. The ratios at which it moves these things has to do with the number of binding sites. The sodium-potassium pump has 3 sodium binding sites, has 2 potassium binding sites, and has 1 binding site for the ATP. So if you are in the habit, like almost everyone is, of saying sodium before potassium when you say sodium-potassium pump and not saying potassium-sodium, I don't know anybody who says that. Sodium-potassium pump, if you call it the 3, 2, 1 sodium-potassium pump, you get 3 sodiums, 2 potassiums, 1 ATP for the pump every time. 3, 2, 1. That makes sense? 3, 2, 1 sodium-potassium pump. So, and we'll figure out why those ratios occur a little bit later, but just stick with me for right now and let us. Okay, you ready to watch it?
Yeah.
Okay. All right, so the sodium-potassium pump, here's a video of it. So I'll bet you can figure out, just by looking, what these binding sites are for. Sodium, and these, and this, ATP, exactly. So what is going to happen is it's not actually going to move sodium and potassium simultaneously, but it's going to be coupled. Because after I dump sodium, I will expose my potassium binding sites. By the way, how did I get the energy to pop sodium the opposite direction that it wants to go? Yeah, I popped a phosphate off of that ATP to do that, right? But now I've exposed potassium binding sites, right? And although they're not simultaneous, they're coupled because I could never use this pump again unless I moved potassium in, right? It would be stuck there forever, right? So now I bind potassium. How do I get it to move? That phosphate falls off, and then I move potassium back in, right? So sodium out, potassium in. For the rest of your life, every single second, you should be tired. That's the reason you're tired, right there, right? It's not parenting. It's like, man, I am busy all the damn time, even when I'm just sitting there watching Mindhunter, whatever. Does that make sense? So this is the sodium-potassium pump. So here's my challenge to you. So see if this makes sense to you. You could build this entire story with a little practice, just starting with this and this. Could you not? If you knew where sodium and potassium were supposed to be high and low, yes, you could easily put in the channels and figure out which direction it would diffuse, yes. And then you could put in the pump and figure out which direction you needed to pump it to fix that problem, right? So the good physiology student doesn't memorize this, they build it. Right? You start with just the concentration gradients, that's homeostasis, that's what I want, right? And negative feedback says that if I mess it up, I better put it back, right? Stick in the channels, jack it up, stick in the pump and fix it, right? Okay, and this will end up being really, really important to understand all of nervous system physiology because as she said, you are going to have to know how this is related to, for instance, action potentials. And eventually we'll figure out what would happen if the damn pump stopped working, right? And your cells will eventually die if the pump stops working. Okay, so we will stop there for today. Next time we will actually talk about the impact on the electrical nature of the cell. Okay? Okay. It's some of them. It's multiple choice, but there's always going to be on the first test. It's really essay on the test is on the bonus. So
Yes,
you
they're
can
kind of
collect
on
the
video.
and then on all the other tests, there's also the essay on that.
Oh,
I just take
How long is it? Entire series. 60-ish questions, whatever. I try to write it so that you're not rushed, but also you're not late. Right?
Essay, how long are they usually?
Did you get like these
You can bullet points. I don't need you to be essays, right? I don't get the flow
How are you
chart
feeling
you want
about it?
on your stuff to find
Well,
it.
I studied last night and I felt good. The only thing I want to make sure I know is it starts with the D.
dimensional and interdimensional